So we're doing lecture 9 today. I'll try to finish it next Tuesday. I don't think I can, but we'll see how it goes. We probably have to cut some of the contents from lecture 10, so you don't have to study as much for the final test. But this is a difficult one, okay? It's even more difficult than number 8 in terms of understanding some of the concepts. So, pay extra attention and do stop me if you need clarification. Biotechnology, what is it? Well, as the name implies, it is technology that's based on biology. Up to this point of the course, we have learned about various biological processes. We learned about how DNA are being copied. We learned about how cell divides. We know how proteins are produced, right? Decoding the codons, creating the amino acids, and so on and so forth. So biotechnology is about trying to control some of these processes, trying to alter them in a way that's going to create a product that will benefit human in one way or the other. It's going to help improve our lives. Okay, so there are a lot of different types of biotechnologies. We're not going to be able to go through all of it. It's a course on its own. But I've chosen a few common ones to go through with you. We're going to be starting with recombinant DNA, and that's probably going to take us almost the whole class, right? Recombinant DNA is DNA that consists of genetic information from multiple sources. So in this case, I don't know if you can see this picture, but these are mice at the top here, and you can see they are glowing in the dark. Okay, so what happens is they took a gene from jellyfish, right? The gene that lets jellyfish glow. It's called the GFP, green fluorescent protein, and then they incorporate it into the mouse DNA. And in the end, you create these glow-in-the-dark mice. They are actually quite useful in terms of research. If you want to know more about it, you can definitely search it up yourself. And we're going to be looking at how recombinant DNA are useful in two contexts here. Okay, first, we can apply recombinant DNA in medicine, and we can also apply it to agriculture. So we're going to look at agriculture as an example first. That's the easier one to understand. Has anybody heard of golden rice at all? Golden rice? No? Okay. So in low-income countries, there is a lack of access to nutritious food in many instances. Okay. And so vitamin A deficiency is often a common condition that people experience. Okay. And vitamin A is actually very important for various reasons, including the production of a special pigment that allows you to see at night, with your eyes. And so people with vitamin A deficiency often have nighttime blindness. And if it continues to happen, then it could actually lead to irreversible blindness. Okay, so this is particularly a problem for kids. Right? Also, vitamin A is vital to having a healthy immune system. So for people with vitamin A deficiency, they have low immunity. And again, in countries, low-income countries, often they have poor healthcare, poor infrastructures, and low vaccination rate. So combining this with lower immunity as a result of vitamin A deficiency is going to create a very severe health crisis, okay? And that could increase the risk of having a disease outbreak, right? An endemic. Right? We all know what a pandemic is now, okay? So at a smaller scale, it would be an epidemic, and then on an even smaller scale, it's called an endemic, okay? So it is a serious condition. It's estimated to kill 670,000 children under the age of 5 and causes an additional 500,000 cases of preventable childhood blindness. Okay, so what can we do? Oh, before that, this is a map that shows you where vitamin A deficiency is quite prevalent in the world. Green is not a concern, so not so much in Canada and US. These are high income countries. And then it's more severe in, like I said, developing countries and low income countries. So what can you do? Well, if vitamin A deficiency is a problem, then it seems like the straightforward thing to do is to give them vitamin A, right? Okay. But it's not always easy to transport these food to these countries in large quantity because of things like price. And even if you're able to get them there, you might not be able to preserve the food long enough to give access to everybody. Okay. And that's where golden rice comes in. Okay? So in those countries where vitamin A deficiency, rice is often a staple food. Okay? It's part of people's everyday diet anyways. And so what scientists and researchers have done is they've incorporated a gene into, two genes actually, into a regular rice and caused them to produce beta carotene. Okay? What kind of food, everyday common food, contains beta carotene? What does your parents tell you to eat because it's good for your eyes?
Carrots.
That's right. So carrots as beta carotene, and it's true. Beta carotene is a building block to make vitamin A. Rice naturally just does not produce beta carotene, but these genetically engineered rice will produce the beta carotene. That's why they are orange. Okay. So rice is a lot easier to transport, and they could be kept for longer periods of time, right? As long as you keep them dry, right? And it's relatively easy to grow, which makes it an ideal solution for the problem. So how do they do it? Well, long story short, they took a gene from daffodils, which are these flowers here, okay? The gene is called Psy, PSY, doesn't really matter. You don't have to know it. Change the language. No, that's not the one I want. And then they took another gene from a soil bacteria called CRTL. They usually write the genes in italics. And they put both of these into regular rice. And that's how they create the golden rice. And now you're able to, fix the problem of vitamin A deficiency. Okay. But as with a lot of biotechnologies and these things, people call them GMOs, right? Genetically modified organisms. There are proponents for it, and there are also opponents for it. So the people who are supporting the use of golden rice is, as you can imagine, arguing that they would help address vitamin A deficiency, right? And that's a good thing because you improve people's health, you prevent people from going blind, right? Especially in kids. And if you improve the immunity, you can improve public health. Improving public health will decrease healthcare costs, and generally, it's a good thing. Okay, and also enhance food security. Right? People now have access to food that are, nutrient rich. But people who oppose this idea, they would argue there might be unintended consequences. Right? Anytime you create a new genetically modified organism, you kind of don't know what the long-term effects is going to be. Like, what if these crops get into the wild? Is it going to become an invasive species? Is it going to affect the growth of other plants, right? Long term, we don't really know. Immediately, sure, it will do all these things, but it's hard to tell what's going to happen in 10 years, 20 years, right? They are also worried about corporate control and patents, right? If the corporation has too many controls over local policies because they're supplying a lot of these food, then that could, affect people in a way that's biased, right? And perhaps it's going to lead to intensify certain social economical factors. Okay, so some controversies for you. Anyways, the key point is this is an example of recombinant DNA being used in agriculture to address a health problem, right? What's recombinant DNA again? Taken from different organisms, okay? So you can take it from the flowers, the bacteria, and put it in the rice. Now you have 3 different sources of DNA in the same organism. That's recombinant DNA, Okay, a lot of GMOs are recombinant organisms. Yes.
Please know specifics on golden rice stuff.
General idea is good. Yeah. Okay. So the other application, which is going to take the whole class, is medicine. Okay. Application of the recombinant DNA in medicine. So diabetes, there are different types of diabetes, type 1, type 2. Type 1 diabetes happen usually earlier in life, and the body cannot make insulin. Okay, so insulin is a hormone that's produced by your pancreas, and it helps you lower blood sugar after a meal. And so if you just had lunch before coming to class, your body is pumping out insulin to help you absorb those sugar into your cells. Okay, so people with diabetes, type 1 diabetes, they cannot make insulin, and then it's a life-threatening situation. The only way to help them is to have insulin injection. Okay, so prior to the late 20th century, the only way to get insulin is to extract them from cows and pigs, and from the spleen of the cows and the pigs. Okay, so of course that's could be expensive. People are worried about contaminants. Okay, if you're extracting from animals, like you really need to sterilize it and make sure that it's safe for injection. And also, the animal version of insulin might not necessarily be compatible. Well, I mean, it's compatible, but then it's not going to be the exact same one as the human ones. So there is concern for efficacy. Okay, it's not as effective as the human versions. Okay, but that was all they could do at the time. And actually, the work was done primarily by these two Canadian scientists, Benting and Best. Okay, and then later on, with the invention of biotech and recombinant DNA, people were able to essentially turn bacteria into insulin factories. Okay, so we are able to basically hijack the bacteria's own cellular processes and kind of force them to produce human insulin. And then once that was done, you are now able to create insulin at a large scale and at a much lower cost and higher purity as well. So we're gonna go through the process of how to produce the insulin using bacteria. Okay, and this is the part that you have to wake up and pay attention to. Right, there's a lot of stuff going on here. 4 steps are involved in turning the bacteria into insulin factories. Number 1, we have to locate the gene of interest. We have to find out, within our 23 pairs of chromosomes, where is the gene for producing insulin. Once we found that, we have to clone it. We're going to make millions of copies of that gene. We're going to pop it into a vector to deliver it to a bacteria, and then nature will do the rest. Okay? So hopefully today we can go through all of these steps. Okay, I don't know. We'll see. If I'm talking too fast, just let me know. All right, first step, locating the gene. Finding out where the gene is located in the genome used to be a very difficult task, right? But all that changed since the completion of the Human Genome Project. Okay? The Human Genome Project was an international collaborative effort with the goal to map out the entire human genome. They want to know every ACTGs that makes up every single chromosome. Okay, and that was a very huge, monumental task that started in October 1st, 1990. Right, and it took you can do the math here, right? It took over 20 years to complete. Right, it was not done until April 2003. And I remember when I was in high school taking biology, we had to do like a research project and presentation. I did my research project on the Human Genome Project. So that was like really long time ago, right? And now, of course, it's completed. It has been completed for more than 20 years. And what happens is all that information is publicly available online. So if you want to find out the location of a gene, all you have to do is go on the internet and you can find it. You can find it. All right, let me see if the internet is working today. Oh, there we go. Okay, so let's go back to the home page here. Okay, and on the home page here, we go to the genome browser. And if you fancy looking up genes for mouse, rat, zebrafish, whatever, and it's all there. But today we're going to be focused on the human. Do you remember any genes that we've learned in the course? No? What was it? I think you mouthed it right. What was it? I don't know. I thought you said the right answer. What genes do you remember from this course? We learn a lot of genes, but then like there's one that maybe you remember about cancer. Yeah, that one. Yeah, what is it called? No, that's okay. That's the usual response. It's the BRCA1 gene, right? Remember? Yeah, so like if I want to look up the BRCA, I type in BRCA1, and then it tells me right away the BRCA1 gene in human is found on chromosome number 17. Okay? And it is nucleotide 43 million blah 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 to 43 million blah blah blah. Right, so you know exactly where it is. Okay? The information is right there. This is the centromere of chromosome 17, right? This is the shorter arm, which we call P. This is Q, and it's right there. Okay? Then what you can do is you can click onto the gene. I mean, I'm just showing you. I don't expect you to, do this for fun because it's not fun, right? And you can click on the genomic sequence. Okay. And I'm just going to click submit. And there it is. That's the gene. That's the gene, everybody. Okay. It's just a bunch of ACTGs. Okay. That's for one gene. Okay. And how many genes do we have? A lot, okay? We have a lot of genes, but all of these genes are going to be on our chromosome. So everybody, all scientists doing research on genes, genomes, genetic research, can have access to this information. All you need to know is the name of gene you're looking for, then you can find out exactly where it is, okay? So that is step number 1. We located the gene, okay? And you know what? You have the skill to change this into the protein, right? Because look, the TAC, you can convert it into, AUG, and then you can look it up into and the codon table, and it's everything we've learned, basically. Okay? So we've located the gene. What's the next step again? Sorry? What does the note say? We're going to clone it. That's right. We're going to clone the thing. Okay? So cloning is essentially making copies of the gene, right? And again, this used to be a very lengthy process. It's gonna take a long time. But since the invention of PCR, you are able to make millions of copies in less than 2 hours. Okay, and PCR stands for polymerase chain reaction. Okay? You might have heard of PCR during the pandemic when people needed to do COVID testing, right? They have to send the sample to do a PCR test and to see if you have the virus genes or not, okay? But PCR has been around for quite some time, way before the pandemic was here, yeah? So anyways, what does it do and how does it work? Basically, this is a PCR machine. It's quite expensive. It's cost as much as a car, but it's quite useful in the sense that you can clone any genes in a short period of time. And so this is where you will put your sample, and you can program the machine to make copies for you. So let's just do a little bit of math, okay? Let's say you start with 1 copy, and that's really rare. Like you rarely start with a single copy of the DNA, okay? You usually have much more than that. But whatever, let's say you start with 1 copy. After 1 cycle of copying, you will have 2 copies, right? And then after another round, how many do you have? Keep going. 8, 16, so on and so forth, right? It doesn't sound like a lot, right? 1, 2, talking about 16 copies, right? I thought you were talking about millions of copies. But let's do this for 30 cycles. 30 cycles, okay? So anybody have a calculator? Yeah. I do too, I think. Let's see. Yep, so right here we can do 2 to the exponent of 30. So that's over 1 billion copies if you do it 30 times. Okay. And how long does it take to do PCR for 30 times? A little under 2 hours. Right. So it's a very efficient way of cloning your DNA. All right. So how does it actually work? The PCR process is very similar to how DNA replicate itself in a cell. We learned in lecture 6 that in cells, we have to separate the DNA strand using which two enzymes? I'm kind of blocking the answer. What enzymes do we need to separate the DNA? What is it? The gyrase. That's what we need, the gyrase, and then we need the helicase, right? Okay. But there is no gyrase or helicase here in PCR. Instead, we're going to be using heat as a way to separate the DNA. The DNA strands are only held together by hydrogen bonds, and they are quite weak, right? Once you heat it up to around 95 degrees, you will be able to separate the strand. The exact temperature, it doesn't really matter. The key thing to know is you use heat to separate them instead of actual enzymes. Right? Then the next thing we have to do is we have to design primers. Okay, so what are primers? Primers are basically the same as the one created by the primates. It tells the polymerase where to start copying. Okay? If you supply the entire genome, you don't want to be cloning the entire genome. It's going to take too long and it's going to take too much resources. You only want to clone the parts that you're interested in. You only want to clone, for example, our insulin gene, which is located on chromosome whatever, right? Okay. So it's kind of like this. You can think of the primer like, let's say I have a textbook here and I want you to photocopy some pages for me. But I worry you forget what page. So what do? I will put a sticky tag here and I will put a sticky tag here. So now I have two sticky tags, right? And I give you the book, you copy all the pages between the sticky tags. Okay? The sticky tags are like the primers. So you're thinking, hey, how do I know what primer to design, right? Like, how do I create these primers? Well, don't forget, you actually have the entire gene available to you right here. So all you need to do is go to the beginning of a gene and you create a primer that's complementary to this section, okay? And then you go to the end of the gene and then you will create a primer that's complementary to the end, okay? So now you have marked essentially the beginning and the end of the gene, and you'll be copying everything between the primers. Do you understand that? All right. One last thing. Normally, in a cell, we would be using DNA polymerase 3 to copy the DNA. But don't forget, in PCR, we have to heat up the sample all the way to 95 degrees, and that is going to destroy the proteins. It's too hot. It's going to cause the DNA polymerase 3 to unravel, and it's not going to function anymore. Right? So to fix that problem, they have actually identify bacteria that lives in hot springs. Right? These springs can go up to hundreds of degrees, and the bacteria that lives there kind of thrive. And so they took the polymerase from those bacteria. They are called tag polymerase, and these are heat resistant and therefore able to copy the DNA even though we heat up the sample to 95 degrees. So that's the process of PCR. Essentially, we are going to separate the template using heat. The primers will bind to it, and then the tag polymerase will know where to copy. Okay, so it will copy this portion, and it will copy this portion. And after a cycle, like I said, you will have 2 copies. So there are 3 steps right here. Let's write it down. Denaturation is using heat. To separate parent strand. So that's step 1 right here. The 95 degree is gonna break the bonds between the DNA strands and they will separate. The next step is called annealing, which cools a little to allow primers to attach. So the primers will find the complementary section and it will bind to it. It's like the sticky tags I was telling you about. Okay, so this is annealing, right, around 68 degrees. You don't want to cool it all the way back down to room temperature, because if you did, then these two strands are just going to come back together, right? So we want to cool it just enough to let the primers bind to. And how much do you have to cool it to? There's actually a formula that you can use depending on how long your primer is, but don't worry about it. And finally, step 3, we're gonna heat it up a little bit. Again, not all the way back to 95, because if you heat it all the way back to 95, everything would just fall off. But you want to heat it up a little bit, just enough to activate tag polymerase. And remember, these are hot spring bacteria polymerase. They normally operate in upwards of hundreds of degrees. So if it's too cold, if it's like 60 something degrees, it might not be doing anything. So you want to heat it up just enough to allow them to do their job. And now you have 2 copies, right? Then you just repeat the process. And you heat it up again so they separate, and then the binding of the primer, and so on and so forth. So this reaction will continue to happen as long as you have nucleotides in the reaction tube. Once you run out of nucleotides, it's done. You know, go on any further. The primers are fine. Primers are recyclable, right? Because you're not actually consuming them in the process. All right. So the key thing to know here is, what's the purpose of PCR? Okay, and what is the purpose of it? Like, what are we trying to do using PCR? Clone, that's right. We use PCR to clone, to make copies, okay? That's the key thing to know. And it involves, 3 steps. Don't worry, I'll give you the review sheet and you'll be fine. Right? And besides, you have to worry about quiz 4 before worrying about test 2, right? Okay, so we located the gene of interest, we cloned the DNA, we have millions of copies of these DNA now, and how much do you think a million copies of DNA is, like in terms of volume? Is it like a water bottle? Is it like the tiny beakers? Like, how much do you think it is? Yeah, it's like it fits in little tubes like this, okay? They are called Eppendorf tubes. It's like this right here. And these are called Eppendorf tubes, and they are this small. And that would be like a lot of DNA. Usually the DNA is just like tiny portion at the bottom. I'll show you a video later. Okay. So I'm just telling you so that you have a perspective of what it looks like. Okay, next is to insert the DNA into the vector. Now I'm gonna slow down because this is the difficult part. Okay, but hopefully so far you're with me. But first, concept check. What do you think? Yes, so quick. By the way, if you want a calculator for Tuesday's quiz, feel free to bring one, okay? Is this the what? 2 to the power 4? Well, it's every time you only double, right? What is it?
Why did you say that? Because I can do that. I don't know.
What is it?
But I know it's 10.
Maybe. Okay, let's say we start with, okay. Yeah, let's say we start with 1 copy, guys. Okay, help me with the math, please. If you start with 1 copy, after 1 cycle, you have 2, right? Okay, and then another cycle, we have another cycle,
8. So that's what? That's 1 cycle, 2 cycle, 3 cycles, and then 1 more cycle, it's 16, right? Okay. But that's if you start with 1 copy. How many copies are we starting with? 10. 160. Okay. Okay, let's try again. You start with 10. After 1 cycle, how many, guys? Huh? 20. And then? So on and so forth. Okay. Now it makes sense. Okay. So basically it's like this. Starting number, multiplied by 2 to the n, okay? Where n is the number of cycles. Okay? Starting number, multiplied by 2 to the n. Okay, write it down. Okay, let's just do one more, okay? What if you start with 20 copies and then you do 5 cycles? Huh? 320? You think so? Huh? 640? Okay, anyone else? But yeah, 5 cycles. 640? Yeah? Yes? You're trying to do it in your head, but you have a calculator right in front of you. 20 times 2 to the 5. That's 20 times 32, which is 640, okay? All right. I'm going to skip this. It's not going to be on the test. We don't have time to talk about this. All right. Next, we're going to talk about plasmid. I feel like I told you this before a long time ago, but let me refresh your memory. This is a bacteria. They are prokaryotes. Okay? We are eukaryotes. That's right. So they don't have a nucleus. So the DNA is just kind of, in the center area. But in addition to their core DNA, bacteria also have plasmids. Okay? And plasmids are circular DNA that gives them some kind of special property. Okay? The plasmid might give them an extra protective shield. I call them capsule. Sometimes the plasmid might make them resistant to an antibiotic. Okay? So like if they have like an ampicillin resistant plasmid, then you cannot kill them with penicillin, for example. Okay. So plasmid usually give them some kind of survival advantage, which is why whenever they have a chance, bacteria will exchange plasmid with each other. Okay. Or if there are plasmid in their surroundings, they might take it up, they might absorb it. Okay. That's the first thing to understand. Okay. So if I'm a bacteria just walking around and then I see a plasmid there, I will likely take it up and put it into my collection. Okay. What does it do for me? I don't know, but let's find out. Let's absorb it first and then find out. If it kills me, it kills me. But like I will take it up and see if it gives me an advantage. Okay, so we understand what a plasmid is. Just to recap quickly, okay? We located the gene of interest. We've cloned it. What's the next step? That's right. We're going to take these millions of copies of a gene and we're going to put it into a vector. And the vector we'll be using would be the plasmid. Okay, so if we can put the gene into the plasmid and we feed the plasmid to the bacteria, then the bacteria will start making the gene for us. Okay, do you understand that? Look, I have a plasmid with the insulin gene. And then I just put it in the environment for the bacteria. They will take up the plasmid, and now they will start turning the insulin gene into insulin proteins, right? And then we can harness them. Okay, so that's the idea. But how do we put the gene into the plasmid? Right, that's the next part. All right. Even though bacteria likes to pick up plasmid from the environment if they can, they actually do not like genetic information to be forced into them. Okay. So under what circumstances would genetic information be forced into a bacteria? We've learned this, at the beginning of the second half of the course. Do you remember? What can forcefully inject genetic information into a cell or into a bacteria? And what is that? That is an example of he said bacteriophage, okay? So I'm asking him, that would be an example of a what? A virus, right, guys? Okay. Viruses can inject genetic information into a cell, right? Into a host. Right? A bacteriophage is an example of a virus. So whenever a virus injects genetic information into a bacteria, they actually don't really like that, and they try to cut it out, right? They have these molecular scissors that would cut them out. And these molecular scissors, we call them restriction enzymes. Okay, so that's the key thing to know. Restriction enzymes are defense proteins used by bacteria to cut up foreign genetic information. They're not going to cut up the plasmid. They're not going to cut up their own DNA. They have a way to distinguish what belongs to them and what does not. So anytime there are foreign stuff coming in, they will cut them up. Now, they don't just randomly cut up the genetic information. Okay? They usually would identify special sequences that they recognize before cutting it. And yeah.
That a sentence, I was trying to read it. It was restriction enzyme, they defend
the bacteria from foreign genetic information. Okay. So for example, genetic information injected by a virus, that would be foreign. That's right. Yeah. Okay, so now they don't cut randomly, they cut at specific sites. Okay, these sites we call them recognition sites, and each restriction enzyme has its own recognition site. I'll show you what that looks like in a bit. Usually, these recognition sites are characterized by complementary palindromic sequence. Okay. Does anybody know what a palindrome is in English? Like Anna, exactly, right? When the word spells the same forward and backward, that's a palindrome in English, okay? Like madam, right? Level. These spells the same forward and backwards, right? Okay. So in DNA, you can have what's called a complementary palindromic sequence. This is the important part, okay? So if you read it forward, GAATTC, right, the complementary strand is the same in reverse. It's a little bit different than the English palindrome, right? It's not like I read it this way, it's same as this way. Okay, please take a look. I'm saying when you read it this way, it is the same backwards on the other strand, right? On the complementary strand. You understand that? And so this is called a complementary palindromic sequence. Right? You don't understand? No, that's okay. So GAATTC, you see it's the same backwards. So that's called a complementary palindromic sequence. Okay, I'll give you an example where it is not palindromic sequence. Okay, and of course nothing ever works. This is not a complementary palindrome. Okay, this is not a complementary palindrome. Even though if you read it this way, it's ACCCCA, and then you read it backwards, it's also ACCCCA, right? But it's not complementary palindrome because when you write a complementary strand, help me out, guys. What's the complementary strand? It's not complementary palindrome. Because if you read it this way, it's not the same on the other side. You understand? Okay. Let's practice creating these things. Okay. I want you to create your own palindrome for me. Okay. Six nucleotides long. You know what? I'll just tell you how to do it. Okay, it's probably really fast. Sorry. Wait a bit around. Give me three letters. Any. Go. Any three.
What? Oh.
Give me three.
Letters?
Three nucleotides. Any.
E, A,
No, nucleotides.
A,
A,
E.
D, C. Okay. So look, this is how you do it. If I want you to create six nucleotides palindrome, you start with any letters that you like. Okay? Watch this, okay? So you know how to do it. ATC. And then on the opposite strand, you are gonna write ATC backwards.
Yeah.
Okay? And then you just fill in the rest. Okay, A goes with and then same thing. C goes with G and A.
Oh. Oh, but
You
you
see
just
that?
have to, like, you don't start it right underneath.
That's right. Okay, you got it? Yeah. One more time, guys. Okay. Give me three, whatever you want. There's no U though.
TPZ.
TTC, okay? Everybody loves the TTC. If there's anything, they're on time. So you put TTC, and then what are we gonna do next?
I think.
Over here, right? But backwards. Understand? Then you just fill in the rest by using base pairing. Okay, so now it's the same this way as this way. Understand? If I want 8 instead of 6, how many letters are you gonna come up with first? 4. Now we're talking. Okay? Huh? Oh, you cannot do odd numbers, yeah. All right. So back to my point, these molecular scissors will find the recognition site and then they will cut it. Okay. Sometimes they will cut zigzag, sometimes they will cut straight down. Right? I'll show you examples. Here is a table that shows you the name of the enzymes and where they are from. Okay. You will hear me pronounce them, okay? But if you want to say them, you just pronounce the italics part first, and then you just read out the regular letters. So this one is equal R1. Equal R1. So equal R1 is taken from E. coli. Okay. And how does equal R1 cut? They will cut by finding these kind of sequences, and then they will cut zigzag. Okay. We have another one here. It's a small one taken from this bacteria. And this one recognized GGGCCC, but it will cut straight down as opposed to zigzag. Okay. So anytime we cut zigzags, like these ones, this one, these ones, okay, these create sticky ends. Sticky ends. Okay. Meaning, even after you cut them, this could stick back together. They could reconnect because the AATT is attracted to the TTAA, right? They're complementary. So they would have a chance of coming back together, sticking back together. That's why we call them sticky ends. If you cut it like this, these are called blunt ends. Blunt ends. And they cannot stick back together. Okay. Yeah.
Oh.
All right.
You have to, like, remember which.
No. I'll always give you this table if I need you to use it.
Oh.
Okay. Yeah. What was I gonna say? For the purpose of inserting a gene into a plasmid, we want to create sticky end. Okay. For the purpose of inserting a gene into a plasmid, we want sticky end. Okay. So these ones are not very useful for us in this application, right? But these ones are. Okay. And I'll show you how that happens. you have a question?
What's the difference?
The difference is this one, you cut zigzag, and then now we have like a hanging end that could come back together. This one cut straight down. There is no chance of putting it back together. Okay. All right. Here we go. And this is what you need to do on the test. I have a sequence right here, and then I have small one, which is a restriction enzyme, okay? Restriction enzyme will recognize this particular sequence. So how many times will the restriction enzyme cut this sample? Now pay attention to the 5' and the 3', okay? And so here's 5', and I'm looking for GGG, Okay, so starting at the 5', okay, just tell me to stop when I hit it, okay? There you go. GGG, right? Cut it. So easy. Do you see another location for cutting? Okay, so again, stop me. Right, GGG, CCC. Gonna cut it again. Sorry, where is it? There it is. Okay, so cut it 2 times. You understand? How many pieces? How many fragments? 3 fragments, right? Okay. You have 1 piece, you cut it twice, you get 1, 2, 3 fragments. Do you understand? Yeah? So? Question. What's the question? You basically just look for this sequence, and if you see it, that's where the cut is gonna be happening. And we'll do another practice. Okay, this one is harder. Okay, I'll let you try first. Right? There are several answers here. Identify the palindromic sequence. It's really hard if you're just looking at the screen. Okay, like maybe you guys didn't print your notes. Test. Please print your notes next semester. Okay, it's very important that you do. You found it? So they should be, 6 to 8 long. Okay. So let's see over here, maybe this one. C T T A A G. Okay. That's a palindrome right here. Do you see that? It's the same forward as backwards, right? Okay. GAATTC. If you read it backwards, also GAATTC. Do you have another one? This one? Oh, this one. Okay, yeah, let's see. Yeah, there you go. That's a nice one. Huh? You see? Same forward as backward. So on the test situation, probably it's easiest if you just write the complementary strand out and then compare it, right? Yeah. But I won't give you something so long. Okay? It would be something much shorter. Okay, take a picture if you need to. I'm gonna move on. Okay, one more example. We have two restriction enzymes now, equal R1 and Hindi3. They both recognize different things. Okay, that's a recognition sequence. So I'm treating this with both restriction enzymes. So you have to tell me how many times it cuts. Let's do equal R1 first. Two times, yeah. Anyone else? For equal R1. Like I said, you will have to do this on the test. Just equal R1 though. Yeah? I don't know, is it? Okay, so what are we looking for? GAA-TTC. So there you go, GAA-TTC, right? So that's going to be one cut. Okay, it's going to cut zigzag, right? Okay, one cut. Do we have another GAA-TTC? Yeah, right here, right? That's two cuts. And that's it for equal R1, right? There's nothing else. But what about Hindi3? So this one is AAG-CTT, which is right here, right? There you go. What's the question? Anna?
Oh, no,
No, you
Just-
don't.
thinking. Thought that would try.
Good. Any questions? Sure.
So for the other example of this thing. It cut straight down because that's how it,
Because that's what they say, right?
Exactly.
You just follow, yeah. If they don't tell you, you wouldn't know whether it's straight or zigzag. So how many fragments here?
four.
Four. Okay, three cuts, four fragments, right? Okay, we're gonna write a note here. Look something like this, okay? Look something like that. Okay, so this here is a sample plasmid. Sample plasmid. That's just the name of the plasmid in the middle. Okay. And I want to talk about these things on the outside. Okay. So these yellow stuff, these are recognition site. Remember, plasmid is genetic information. It's DNA. It's just circular. So if you have GGATTC here, for example, that's gonna be a recognition site for equal R1. So if you treat the plasmid with equal R1, it will cut right here. It will pop open the plasmid. If you treat it with BAMH1, it will cut right here. So basically, it tells you where the cutting sites are if you use different restriction enzymes. Okay, the stuff on the inside, these are antibiotic resistant genes. Okay, they are used as selection markers to distinguish, to tell apart, okay, to distinguish bacteria that have accepted the plasmid from those that have not. And remember, I can provide the plasmid to everyone, but not all of them will take up the plasmid. Whether the bacteria has taken up the plasmid or not, they will look exactly the same. There's no way to tell them apart visually. So I need a way to distinguish them. And one way, there are different ways to do it. One way to do it is to include an antibiotic resistant gene in the plasmid. And I'll tell you how that works in a bit. Why don't we take a quick break? You guys look like you need one. Let's resume at 1:35. All right. So over here, let's say we have a little tube. And in this tube, we have plasmid. You can buy plasmid online.
Wait,
Yeah, this is so I'm gonna draw a plasmid here. And then I'm gonna put a recognition site here. So there we go. I'm gonna have a recognition site here. I just made this up, okay? I'm gonna say this is recognition site. Recognition site for restriction enzyme 1. Yeah. So there's some hypothetical restriction enzyme, and it will recognize this AAA-TTT. And then over here, in a separate tube, I have the gene of interest. Millions of copies, right, of the gene of interest. And how do I get this? From PCR. Okay, so if I want to clone the insulin gene, I just take a sample from any human, a cheek swab or something. I prepare the primers, and then I can clone the insulin gene. Okay, so like I'm gonna draw the gene out here for us. Okay, I'm gonna color it blue so we can track it. Okay, this is the gene of interest. Okay. Now, this is an oversimplification, but hopefully it will convey the point for you. So The gene of interest should be flanked by the same recognition site as the one in the plasmid. So we have the recognition site in the plasmid. You want to have the same one that's flanking the gene. You don't want it to be inside the gene, because then when you start to cut them, you might end up cutting the gene in half. Okay? But you want it to be flanked, like on both sides. Yeah.
What we are doing.
We are trying to put the gene in the plasmid. Okay. Yeah. Yes. So this is the plasmid, right? Okay. We're gonna feed this to the bacteria, but before we feed it to the bacteria, we want to put the insulin gene in it, so that when the bacteria take the plasmid, it will make the insulin for us. Okay. So remember, we already copied the insulin, right? But we cannot just feed the insulin gene to the bacteria. They're not gonna take it up. Okay. You have to put it in a plasmid. If you just feed the insulin gene into the bacteria, they will cut it up anyways, right? Okay. But if you put it in a plasmid and then feed the plasmid, then it's okay because bacteria are used to plasmids. Right? Otherwise, if you put this in, it's like putting a virus DNA in, it will just chop them up. Okay. Now I'm gonna put something else here as well, just a little bit right here. All right, so this portion is ampicillin resistant gene. When you look back next semester at these things, you'll be like, oh, so easy. I wish we could do biology on again. There's so much more to do next semester. This would be like Mickey Mouse stuff. Okay. Ampicillin, yeah. It's a type of penicillin. Okay. I'll tell you what it does later. Yeah. Okay. So now I am going to treat me like I'm gonna add, okay. I'm gonna add restriction enzyme 1 to the plasmid. Okay, and then also same thing here. I'm gonna treat the gene with restriction enzyme 1. So what does it do? Well, since the plasmid and the gene both have the recognition site for the restriction enzyme is gonna cut it, right? It's not gonna cut it like randomly. It's only gonna cut it over here, right? Okay. And let's say it's gonna cut it zigzag because I told you earlier we want sticky ends. Okay, so let me try, hold on first. I always mess this part up. But let me try my best here. Okay, so we're gonna cut here. Bless you. Bless you again. Do you see what I've done here? So basically, you expose it with the to the enzyme, and the enzyme will cut it, and now it pops open, right? Pops open. It cuts zigzag. And what do we call these ends? Sticky end, because they can literally stick back together, right? The TAA will bind to here, and the TT will bind to here. Okay. People actually do this in real life. Like if you if you take introductory biochemistry in university, this would be one of the labs that you'll be doing. Okay, how about this one? Well, let me try again. I'm not gonna ask you to draw any of these on test, obviously, but again, I'm hoping this is gonna help you understand. Hey, by the way, please take a moment and try to do the course evaluation when you have time. Okay, let me know what's working for you, what's not working for you. I would love to hear about it. I know, I know. Okay, so what happens is you will cut it, and you've generated complementary sticky ends. Okay, specifically, okay, I'm hoping this is where it all makes sense to you. Look, this piece, if you mix these two things together, this piece can actually stick to where? Left piece or right piece? That's right. Okay, and this piece can actually stick to here, right? So if I put these two things in the same vial, okay, and I add a little bit of glue, what's the enzyme that acts as glue? Do you remember? Starts with L, ligase. That's right. If I put these together in a tube and I add ligase, then this will literally go into the plasmid. Okay. Let me see if I can do this. I don't know if I can. No, it's gonna look horrible, but like you get the idea, right? Yeah. You just put it in. Okay? You just put it. Yeah, don't draw this, okay? Yeah. Yeah, it looks like terrible. Okay, don't forget the ampicillin gene right here. There you go. Sorry. Want higher? Yeah, so remember, this is in one tube and this is in another tube, right? So I can just mix the two tubes together, add ligase, which is the glue, and then this will go into the plasmid. And now I have the plasmid with the gene that wasn't there before, right? Because this part will stick to there and this part will stick to here. Okay. Because look here, the hanging part is AATT, right? Which would bind to the TTAA. Okay, we've located the gene, we've cloned it, we inserted the gene into the vector, which is the plasmid. Then we have to feed it to the bacteria next. Okay. By the way, we are learning how recombinant DNA is being applied in medicine. Okay. Right, how to produce insulin to treat diabetes. Yeah.
Wait, so it's not genetic because my literal entire family has that. Like not
There is a
entirely.
genetic component to it. Yeah. But again, there's type 1 and type 2, right? So, it depends which one we're talking about. You can lose your what?
Like,
Lose your leg. Oh, yeah. It could. Yeah.
but type 1 is just the one where you just
We receive the shot. Yeah. Type 2 don't receive the shots. Yeah. Mhm. Okay, so we're gonna feed it to bacteria. Okay, we have a bunch of bacteria. These are bacteria, okay? Now, you can encourage the bacteria to take up the plasmid by um, putting them on ice a little bit and then putting them into heat, or you can shock them with a little bit of electricity. There are a bunch of stuff you can do to encourage them to take it out. However, even the best approach is not gonna achieve 100% acceptance. Okay? Some will not take it. Okay. It's like, if I brought candies and I let you guys take it, some of you will not take it. Okay. And that's fine. Right? And that's what the bacteria will do. Right. Some will accept it. Right. Some will not. But how do you know? How do you know? Okay. So what's going to happen is here, plasma to the bacteria. And then afterwards, we're going to have this situation. We're going to have this situation right here. Okay. Some of them will have the plasmid. I will use blue circles. Doesn't really matter. Whatever stands out to you, okay? Some of them will have the plasmid. Okay. Some of them will not. So the one that has accepted the plasmid, we call them transformed. Transformed. Okay. Transform means accepted. Accepted the plasmid. Okay. And this one did not. Did not accept the plasmid. There you go. But again, how do you tell? Right? You can't tell by looking at them. Even if you put them under the microscope, you can't tell if they have the plasmid or not. Okay? By the way, which one do we want to keep? The transform one or the non-transform one? Good. Yeah. We want to keep the transform. We don't want to get confused. Okay? Transform ones are the ones with the gene. That's the one that's going to be making the insulin for. I just remember something kind of funny, I guess. I was making this seafood stock, and then so like I have the shrimp and everything, and then I was making it for like half an hour, and it was finally ready. And then I told my daughter to hold the sieve so I can sieve out all the stuff. Right? But I got confused at the moment, so she hold the sieve over the sink, but I didn't put the container down, and then I pour the whole stock down the drain, and then I end up keeping all the scrap that I wanted to make out. Anyways, this reminds me of that story because after all the hard work, we want to keep the transform one and not the empty ones. Okay, we want to keep the good ones. Anyways, yes, it was so sad. I screamed, and she was like, What did I do?" I was like, "No, nothing. It's all me." Okay, so how do you tell? How do you tell? Okay, over here we're gonna have a plate right here. Okay, this is called agar plate. Agar plate. You can Google it to find a nice picture. Plate. Sorry, plate. It's basically something to grow the bacteria on. Okay, so here we have agar play plus nutrients. It's about this big, okay? And it's brown, and it smells a little bit. So what's gonna happen is you're gonna take all these bacteria and you're gonna drop them onto the agar play with the nutrients. You're gonna spread them out and you will let them grow. Okay? So what's gonna happen? Well, if you give them nutrients and you let them grow, and they're bacteria, of course they're gonna grow. Okay? In fact, you're gonna have a lot of growth. Right? All these orange things, they are like bacterial colony. Okay, so you would expect to have lots of growth. Okay, so this is actually a control for the experiment. Okay, what does this tell you? It tells you you did not kill the bacteria because we have been manipulating them, right? We have been, shocking them with electricity, heating them, putting them on ice bath and stuff like that. Like we want to make sure that all those things didn't kill them. So by putting them on an agar plate with nutrient, you would expect lots of growth, and then we know they're alive. But this does not help us distinguish the transformed one from the non-transformed one. In order to do that, we need to have a second plate. Okay, so this second plate is also agar plate. Everything is the same. Agar plate plus nutrients and plus ampicillin, which is the antibiotics. Now, let me remind you, in our plasmid, not only do we have the gene of interest, but there is also the ampicillin-resistant gene. Okay? And this is the important part. If the bacteria has been transformed, if it has accepted the plasmid, they now have resistance for ampicillin. Right? So even if you add ampicillin to the agar plate, those that are transformed will be able to survive because of the ampicillin-resistant gene. Anything that has not accepted the plasmid will not have resistance, and they will all die. Okay? So over here, what do you expect to see? Well, you would expect to see some growth, right? But not lots of growth. Okay? Only transformed bacteria can grow. Okay? Nothing else will be able to grow. And these are the ones we want. These are the ones we can scoop out and then put it on a regular agar plate. Then they will flourish and bloom. And at the same time, they will continuously pump out insulin. Right? And you can collect the insulin from the culture, purify it, and you get a pharmaceutical product. Right? It's not, yeah.
So is there a reasoning why, like, not all the bacteria will will transform?
Yeah, because again, right? Some want to take it up, but in general, they might not like because plasma is pretty big, right? So they might not be able to completely internalize it. Yeah. Yeah. Again, it's like the candy analogy. Some people will take it, some will not. So it's not just insulin you can do this for. Okay, you can do it with other stuff. Right? There's a lot of proteins that we want to produce in large scale, and a lot of time we use this particular technique. Well, that was a lot. Okay, let's try a few more questions here. Okay, so here we have another plasmid. Now you will have to do questions like these on the test, right? Here is another plasmid with different restriction enzyme site. Okay, this is another resistant gene. TET is tetracycline. It doesn't matter what it stands for. It's just another antibiotic. So here they're saying we are inserting a gene using equal R1. Where's the gene gonna go? Right here, right? It's gonna cut open and then pop the gene in. And then you're gonna play all these things on an agar plate with tetracycline. Do you expect all of them to die, all of it to grow, or some of it to grow? Think about the candies. Are all of you gonna take the candies? No. Are all of the bacteria gonna accept the plasmid? No. So only some will grow, right? Yeah, only some. Is that what you said? I'm sorry, it must be the angle. Okay. Okay. Oh, is that what you said too? That's what you said. I'm sorry. 3, right? Yeah. Sorry. Yeah, only some will grow. We got it? Okay, the next one. Pay attention. It looks like the same question, but it's not. Okay. This time we are inserting the gene using which restriction enzyme? Small one, okay? So small one is right here. Yeah. If we insert the gene with small one, it goes right here, right into the middle of the resistant gene. Okay? And that's going to destroy the resistant gene because you interrupted it, right? Okay, with something, you're putting something in it. Right? So what does that mean? This means even if the bacteria accepts the plasmid, do you think they will survive in tetracycline? No, right? Because there's no resistant. The resistant gene was destroyed because of the insertion, right? So what's going to happen to them? All of them will die, okay? If you are not transformed, you're definitely dead. If you're transformed, it's not going to help because there is no resistance. You understand? So that's why a plasmid has multiple insertion sites so that you can use the most appropriate one. Now, on Luminate, I will let you go soon, okay? But please, this is important for you to practice. Someone sent me a message. I wonder who it is. Stop sending me messages, guys. Email me instead. Where is this is it. Number 9. Study resources, transformation and selection markers, okay? If you're confused, you want to read this. All right, so like I give you more examples, okay, but I want to do this with you right now. Over here. Okay. So okay, how do I do this? This is not working. Let me download it first. Maybe it works better that way. Give me a second. Okay, so you can read all this. I typed this up, okay? So it's pretty much the same way I explain it in class. But let's do this one. Okay, let's do this one. Okay, we have a plasma right here. Can you see it or is it too small? Too small? Better? Yeah, hopefully. Okay, so we have a plasma here. We are going to be inserting a gene using ALU1, okay? So, we feed it to the bacteria. Agar plate with nutrients. Lots of growth, no growth, little growth. What's going on? Yeah? What do the rest of you think? Insert a gene using ALU1, put it on agar plate with nutrients. Lots of growth, little growth, or no growth. But is there antibiotics in this plate? No. So are they going to grow or not grow? Lots of growth or little growth? Lots of growth, right? Okay. In fact, anytime it's agar plate with nutrients is always lots of growth because there's no antibiotic to kill them, right? And you give them nutrients, they're going to grow. Okay. What about agar plate with this antibiotic, which is CAM? CAM is right here, right? So do you expect them to have lots of growth, no growth? I forgot what 3 is. What is 3? Little growth, yeah. What do you think? No growth. Okay, why? Because we inserted the gene using ALU1, which goes right into the middle of the CAM resistant gene. So even if they accepted the plasmid, they will all die. Okay? What about ampicillin? Okay. Lots of growth, little growth, no growth. Little growth, that's right. So ampicillin is over here, which is unaffected if you insert over there, right? Okay, so it's not like a lot of growth because only some will accept it, right? Okay. Let's do one more. Okay. You try it out, and then I'll let you go. Okay. I can't show everything on the screen. Okay, just it's hint D3 with the same column. So think about it. Okay, it's the same map. Okay, this time we're using hint D3. So we are using, that's right. Using this. Yeah, with nutrients is always lots of growth. Okay, think about the other two.
Well, yeah, no, let me do the.
Lots or little? Little bit, right? Okay, remember, not everybody accepts it, right? Okay, and if I insert using hint D3, it's not gonna affect the resistant gene for CAM, so it's gonna be okay. They will grow a little bit. Okay, what about ampicillin? Lots of growth or little growth or no growth? Okay, you better understand this. Okay? It's gonna be on the test, final test. Okay. So practice, and then the answers at the back here. Okay. All right, and then there's like tons of explanation here. Good luck with the quiz on Tuesday. Study hard. Next week, yeah. Yeah.